So on the previous slide, you were provided with a YouTube link where I demonstrate enzyme activity and how it works. We talk about what enzymes are made of. Enzymes are made of protein, and they are sensitive to changes in pH and changes in temperature. And we talked about what denaturing meant and active sites and all of that, and the term substrate, etc. So if you have not watched that, please take a moment to watch that, and then what we're going to talk about here is the first experiment that you're going to run for this lab. So experiment 1 of this lab is the effect of time on enzyme activity. And the reaction that you're using is called the hydrolysis of starch. So I wrote out the reaction for you beneath there where you see hydrolysis of starch, you see the term starch, and then you see water, and then you see amylase, and then you see glucose. So let's identify from this reaction, what is what. So it's pretty simple to go ahead and begin by trying to identify what is my enzyme. And so when you look at all those terms, there's a term there that is named in the way in which we name enzymes. And if you recall, we name enzymes by putting ACE on the end. ACE on the end of a term indicates that it is an enzyme. So amylase is the enzyme that we're working with, and the source of the amylase that we're going to use is saliva. So the enzyme amylase in your saliva can be called salivary amylase, indicating that it comes from your saliva. We also have an amylase that's produced by the pancreas, and we call it pancreatic amylase, anyway, just to distinguish between the two sources. But you're going to be using salivary amylase, so that's in your saliva. The substrates of this reaction, so remember that the substrates are whatever the enzyme is acting on. So the substrate is going to be starch and water. These are substrates. So if you remember from a previous chapter where we did the biological molecules, the term hydrolysis is the name of a reaction where hydro indicates water, so that means adding water, which is what we've done here, and lysis means to split. So if we add water to starch and we add an enzyme to catalyze the reaction, we will split the starch. Okay, and what we're going to split the starch into, ultimately, it's going to be split into glucose. So that would make glucose our what? That would make glucose our product, right? So we have our substrates identified, our enzyme identified, and our product identified. So that's the reaction that we're going to watch. Let me also indicate a couple other terms just to remind you from our previous chapters that starch is a big, bulky carbohydrate. Starch is known as a polysaccharide. So it consists of many rings bonded together, kind of like this. And the objective when you are trying to digest something is to break it into its monomers so that then it can be absorbed across the digestive tract. So this polysaccharide is a polymer. It's made of many subunits, and these subunits are all glucose. So each one of these rings is a glucose. So through the process of hydrolysis, we're going to convert that polysaccharide into the monosaccharide known as glucose. So glucose is a carbohydrate that consists of one. I'm going to run out of room, so I'm going to squish it in there. There we go. That says monosaccharide. So glucose, again, is a carbohydrate made of a single ring. So it is known as a monosaccharide. And the objective here again is to take that big bulky starch molecule and digest it or hydrolyze it or split it into it's the products, which would be simple monosaccharides, which would be glucose, that can then be absorbed across the intestinal wall and into the bloodstream so that our cells can then use that nutrient or fuel molecule, if you will, to produce ATP or to string it together as glycogen or a variety of other end fates. So this is the reaction that you're going to be watching. And so where you're going to get your starch from is from the crackers in your kit. So you're going to take the crackers and you're going to use 2 of the cups that you're provided in your kit. And it tells you when you read the instructions for this experiment that you're going to put a C, you're going to write C on 1 cup, that's going to be your control cup. And then you're going to write E on the other cup, which is going to be your experimental cup. And that you're going to crush about half of a saltine cracker into the bottom of the control cup. So there's my crushed up saltine cracker, the bottom of the control cup. Then it tells you to use a 15 ml centrifuge tube to measure 10 ml of tap water and add the water to the control cup. So you've added water now. So in my control cup, I have a smushed up cracker and I have water. Then for your experimental cup, you are going to chew 1/2 of a saltine cracker for at least 30 seconds to 1 minute. Now, if you really want to get this reaction to go, I recommend chewing for 1 minute. So you really get a lot of good saliva in there and you get it mixed up. When you're crunching on that cracker, what you're doing is you're breaking it apart, right? So you're increasing the surface area to give the enzyme more surface area to contact. So if you can stand it, try to chew it for a minute and don't swallow it. You then are going to spit the contents of your mouth. You're going to spit the chewed up cracker with the saliva added to it into your experimental cup. So that's what you're going to have over here. You're going to have the mushy saltine cracker that we have added what to? What have we added to it because we chewed it? We've added the enzyme amylase, right? Because the amylase was in our saliva. So now you have the cracker, and now remember the cracker, and this is also provided for you in the reading as well, but the cracker is mostly starch, right? So the cracker is starch, and you've added the amylase enzyme to it by chewing it in your mouth, right? And so what you're going to do next is it tells you, this is at number 6 in your instructions, that you want to add more saliva to it if you need to so that it equals the volume of what you have in your control cup. So if you need to increase the volume in the experimental cup, then you're going to spit into that cup. Okay. Now, it tells you then immediately after setting up the control and experimental cups, you want to place a glucose strip into each. So you're going to add 1 glucose strip into the control cup, and then you're going to add a second glucose strip into the experimental cup. It tells you then that you're going to leave that strip in place for 30 seconds. You don't want to go longer than that. So you want to time this, and at 30 seconds, take those strips out. Then you allow the color to develop for another 30 seconds. Again, set your timer. You want to let them develop for 30 seconds. Then you're going to compare the color of your glucose strip to the color chart provided with your kit. Once you select the color that it looks like it matches your strip, you're going to record that data or that value into table 1. And you're going to record it underneath the column that's marked 0 minutes. Okay, so that's going to be pretty much your initial reading. What were my initial glucose levels at that time period. Then you're going to repeat steps 7 through 10. So what you're going to do is don't reuse those glucose test strips. Get another one and submerge a glucose test strip into each cup. And you're going to let it sit this time for 15 minutes. So you're going to let those glucose test strips rest in there for 15 minutes. Then you're going to take them out, let the color develop for 30 seconds, and then read your results. And then you're going to record your results in the table, table 1, underneath the column that's marked 15 minutes. And so hopefully you'll see a change with one of those cups, right? Because again, when you put the glucose test strips in there, if it tests negative for glucose, that means glucose is not present in that cup. If it tests positively for glucose, that means glucose is, which means what? It means you must have broken at least some of that starch in the cracker down, right, into glucose, if glucose is present in your cup, and you're using the glucose test strips to determine if glucose is present or not. So from there, once you run that, you're going to see what your results are, and then you're going to answer some questions. So it's a great little experiment, and I hope you have fun doing it. If you have any questions, please, feel free to give me an email or call.